Today what I want to deal with and is really give you an exemplar of the kind of developments and frameworks that we have, our team has developed that are now available and accessible to you. I'm going to use some use cases without diving in really deep into results of each and every project. But the aim and the goal is really to see and give you a feel of what is feasible, what is possible, so that you can see connectivity with the research that you have ongoing in your own lab, or how and where you see your needs sort of emerging. So I'm going to start with a quick introduction. And I will organize this into three different sections, which will be the first part focus more on analytics and the frameworks that are available. The second part, which is focused more on web-based methods and tools that will be there, and also computational and AI methods. And then some of the tool developments that are going to be in the web-based portals. So I think with most everyone here, you have been working with one of these organisms, if not something very similar, right? Viruses, bacteria, microbes, fungi, animals, plants, insects, biomedical side with patients, you name it. And there are many sequenced genomes that are now publicly available. And many of these are used as model systems or things that are really utilizing and providing a lot of information if you are working with any new emerging organisms or even orphan crops for that matter. What I would like to highlight is and stress is the cost of the human genome. If you start about 20 years back, more than 20 years, a quarter of a century, the cost was expensive. You know, the first draft of human genome, it was well over 100 million. And as you can see here, the cost has continued to decrease, bringing it close to $100 per human genome. And this really opens up a lot of potential opportunities for utilizing this as part of the regular clinical care for most from the patient perspective. But what has really happened, it has managed to beat this Moore's law prediction, which was basically predicted at halving it at every 2 years. And you can see we have been well below that for quite a bit of time. Now, this does open up plenty of opportunities for us. You see a lot of studies happening which are utilizing these genomes and going beyond the genomes into this multi-omics data space. And you can see you have everything from epigenome to bulk, transcriptome, proteome, metabolome, phenotypic, depending on if you're working with patients or animal models, or even on the plant science side, the different traits, the different diseases, regulatory aspects, as well as taking your environment into consideration, right? So all of these layers are really telling us about everything that is happening in the same organism, in the same species at different times when you take your time and your dimensionality into consideration. And then you can scale out to different cells, tissues, organelles, even population level, where you're looking at the health of the entire community. What you do want to integrate is the phenome, which in case of patients would be the clinical phenome, which comes a lot from your lab measurements and your measurements in your EHR systems. But also from an Agribiotech perspective, the different traits that maybe some of the crops or the breeders would be focused on. And all of this is very well linked in terms of the environmental space. Now, the global genome sequencing market is really predicted to continue rising, right? Here we are close to where we are right now in the 2024-2026 phase. If you look about 10 years down the line, you're going to go to very close to about 10 times, somewhere in 18 billion range. So what all of this is making us feasible is that the data and the level and the scale of the studies that you can be involved in, but at the same time, informatics, data science, and AI solutions are becoming essential because your amount of information, the amount of data that gets generated and needs to be integrated or looked at simultaneously is continuously growing, right? This is good as well as bad, depending on how you approach it, right? But what has really enabled is the informatics and AI technologies, because now it is preparing us to be able to utilize all of this to make a lot more predictive analytics. And this is applicable to any organisms that you would be working from, everything from viruses, bacteria, all the way to animal and human beings. What has really changed over the period is gone are the days where, similar to this news outlets, like there used to be a few newspapers which would be the source of information. Everyone would be going and, just gathering information or using that as the major source. Whereas now the new model is where in the social media level, everyone is generating data and at the same time, everyone is consuming it. Now, the same is very much true in the case of research, right? Every lab is generating data. These large, big consortiums like all of us, UK Biobank, Kidney Precision Medicine projects, they're all generating huge amounts of data, which would be really fantastic for the labs who are working in that space to use. But at the same time, anything you generate in your lab has to be contributed to a repository. And it's a requirement when you're going and publishing some of the work, right? But this is fantastic because others can use that data. Now, what is making it challenging is that this diversity, right? There are so many different data types, there are so many different repositories, and not everything is deposited at the same place, where it's easily connected to the other pieces that you can quickly find or utilize. They are spread across different repositories, and then these large amounts of comparisons, and the larger the size of the project gets, the more number of comparisons you're looking at, the more complex all of this becomes, right? And this is true depending on whether you're working in agribiotech, like a precision agriculture field, where you're trying to integrate all of this information coming from soil, environment, crop management. You decide about, you want to increase yield or what sort of plant populations or germplasms you should be working with. Are they suitable to grow in certain environments, or how does the economic and marketplace kind of thing play into it if you want to make improvements to the yield or decisions about the crop susceptibility in drought or different perspectives. Now, same is true if you're focusing more on the food security related questions, where the population is doubling by about 2050 to reach about 9.7 billion. And if you are really projecting the growth that you see in these 4 different crops over here, traditional growth would only take you land up to where the solid lines are, whereas the demand is really going to be way more, as shown by your dotted lines. And so how do you get there? How do you tap into germplasm, genoplasm, and the phenotypic things and the studies that have been done so far? Same is true if you're focusing more on viruses, vaccine development, and incorporating genomic epidemiology decisions. And what better example than the pandemic that we have come through, where you can really use information from sequences to come up with a vaccine solution pretty quickly, but also use all of the interactions between genetic and environmental factors, or be able to trace, between different clusters and cohorts when you have this sort of, any new public health concerns, really, what we are going to be facing with. And so a lot of these things are all where genomic information comes at the bottom line, and this is where the technology and informatics pieces become relevant. Our focus, a lot more on the biomedical side is trying to see where we can predict diseases, where we can see whether somebody is, susceptible or has more risk for certain diseases, what the associated genes might be, how can they really explain the molecular mechanisms behind it, how can clinicians tap into this? Because really what you want to make is changes all the way back to the clinical care, and where every patient can get their individualized treatment, and gone are the days of that one size fits all, right? And so we want to make sure that this gets all the way back to our clinicians and not just something that exists as a method. So here I would like to touch upon some examples. So what you can utilize here is, really incorporate information coming from these large tissues or cell types, which a lot of our single cell and spatial omics is now allowing us and enabling us to do, but at the same time, a link all the way from your gut microbiome to linking the imaging data, which is very prevalent and very commonly utilized in radiological interventions on the clinical care, and really bring all of them together to improve your prediction, understanding pathogenesis, coming up with therapeutic solutions for a lot of this. And there are many examples in this realm. So this one is in Crohn's disease, where you can see all the different triggering factors, and you can study the impact of these by integrating the different omics layers, use it to stratify the patient population, understand the links to these individual phenotypes, but at the same time, use the information that's coming from animal models at the bottom over here so that you can design the treatments that are most suitable for each of these individual types. And this is where the whole connectivity with clinical trials and so forth, or the cross-species comparison and translation from animal models to human, starts becoming more important. This has really been leading the way in cancer, where personalized cancer therapy is something that everyone is focused on by using the molecular profiling information from the cancer patients to really predict prognostic markers, predictive, events of whether somebody will respond to immunotherapy or not, and then again use that information to give them customized treatment options. Now, what has enabled us to go there is all of this molecular profiling technology. So you have the transcriptomics, whole genome, whole exome sequencing, and a lot of key market players such as Foundation Medicine, Tempus, Caris, and so forth, who now have panels and can utilize whole genome sequencing, copy number variation predictions, tumor burden calculation, and so forth, so that now you have information that separates out one single patient from the other one and see what is going to work in this patient with much more fewer side effects. So what does all of this information mean, right? You really have to start thinking more from a big data perspective. Now, you are going to have lots of information that needs to be analyzed, that needs to be integrated. You have to come up with solutions for different storage, dashboards, because you want to bring it back to the user, your end user. Computing, where you have to mash all of this information together and apply, machine learning, deep learning, AI type of models. When you're working on biomedical side, you have to be equally, aware of security concerns and really have your systems managed in a way that are really going to support a lot of these things, right? So the things that we really tend to focus on is something that encompasses a bit of each and every one of this component. These things are really allowing us to emerge from that early layers of these multi-omics, but really go more in the direction where you can enable now predictive models. You can do real-time analytics and make decision-making much more seamless, apply these emerging deep learning, AI, large language models, and RAG type of technologies, so that now you're not just simply collecting data and making decisions, going up to the step of analyzing it, but simultaneously building a framework so that you can use information at a population level to really understand what is happening from all of these studies and bring it back to your own patient population. There are plenty of applications in this space, and if you look at farming mainly, you will see people are more focused on predicting yield in crops, monitoring diseases, or doing disease detection in an automated fashion, weather forecasting, and deciding what needs to be grown where. Soil and nutrient information. They're also where they are focusing more on precision farming and how do you use the sustainability component of everything that needs to be applied from a farming perspective. If you're looking at animal science studies, they are again using a lot of AI in understanding the behavioral mechanisms between the animals in a cattle or a beef industry, or environmental management, their growth potential, really their disease detection by automating and using sensors for a lot of these studies. And then on the biomedical side, there are more and more examples where we can use digital health monitoring systems, as well as link them with the personalized treatments, get medical forecasting, and really do a better job of predicting who's at risk or who's going to respond better, and come up with solutions by integrating all of these so that newer drugs and newer therapies can really be designed. So when my team focuses more on enabling translational research, this is really what we are doing on a regular basis. We are starting with this diverse, huge amounts of bulk and single cell or spatial data. We are integrating it with knowledge that is already known in terms of functions, pathways, regulations, sometimes actually predicting new things using AI models now. But in doing so, we also focus more on enabling systems or frameworks where you can integrate all of this data together. And then we work with all kinds of organisms, all the way from animals, viruses, plants, and human diseases. But our goal is ultimately on that application side that I just started with, that we want to bring solutions or tools back so that our collaborators, who are the clinicians, the oncologists, the breeding or the farmers or genomic epidemiologists, are being able to use that to advance science. So when I think of this area, we really have organized that into three essential pillars. And so the first pillar is that high throughput analytics and framework. The second pillar is where we do a lot of web-based tool development so that you can make use of this data seamlessly without having to go to a Linux server to look through millions of records. And then the third piece, which is purely focused on computational method development, which also involves a lot of AI-based solutions as we are going into future. So this is really going to be my blueprint for what I'm going to talk about and show you examples from in the next slides, right? The first section, I'm going to refer to a lot of these frameworks, which are already well published that you will be able to utilize immediately. The second piece, where there are tools developed within these frameworks that can be utilized for some specific purposes. And then the third piece over here, where a lot of computational methods and models have been developed, again, readily publicly available. And this is the space in which we continuously keep developing. Now, the good part is all of these and many of the things I'm going to talk about, you will see citations on it because a lot of this work is published. You will see URLs to the websites on it. That means you can go and access it. And anybody and everybody in the West Virginia INBRE network or otherwise all over the world are able to access these developments. So I'm going to start with our first pillar, and we will start with just few exemplars of what type of analytics we support before we dive into our frameworks component. So generally speaking, any research that is generating whole genome sequencing, epigenome, bulk, single spatial, proteomics, and you name it, right? I don't need to read through this whole list all the way to single cell attack, seq, or so forth. We have pipelines that are developed and are utilized for the analytics component. What you're seeing here is just simple pipelines for bulk or de novo assembly, where you can also integrate long reads or PacBio isoseq reads to build your own genome models for maybe sometimes when that organism or that plant species or animal species is new and you're building your own reference from all of these and annotating your genes. Now, that is just a starting point. And so what you will see me at the end is refer to the tools that this will flow through. Think of your typical examples or your experimental data, right? Any omics data that you tend to generate, you are going to have a need to look at qualities, look at your PCA components to check your replications, do overlaps with your results, look at what is up or down regulated, no matter it's transcriptome, proteome, metabolome, look at your differential perspectives, your clustering, your enrichments, your networks, your regulation part, and the list just goes on, right? A lot of this is very standard. This is very typical of what is the first, cut that you want to make, go and look at the data. For us, many a times, this is not the end point. This is the beginning point for us, right? Because once you have this analyzed data available, we want this data to flow into some of these other tools and methods and frameworks, which is what we have developed over the period, because this is something that is needed. This is commonly needed, and this is what allows the us to take the data more closer to our collaborators who are really have a way to now work with the data and interactively access it, right? Rather than working in an Excel file or with a list. So I will touch upon some of these things as we go, but this will give you a flavor of what is typically involved. If you're working with single cells, you would be familiar. Many a times you would see cell type annotations are going to be your first requirements. You're clustering, breaking it down, identifying the markers for your clusters, doing comparative analytics based on how many conditions you have, looking at differential cell counts between your conditions, or also doing cell communications or regulatory aspects is what you want to do at that first initial cut, right? But then there are things that you can do with your single cell data that you will be able to flow into so that you can then utilize it better for predicting models or getting more into your pathway perspective. So I'll show you examples in a bit. Now, here I want to touch upon one example, which is with our collaboration with Dr. Shirvan and Yoko, and this is more on cancer immunotherapy. So, cancer immunotherapy is kind of a new revolutionized cancer treatment where you are really trying to come up with a new paradigm where non-viral cancers can really have, some molecule or something given similar to a vaccine treatment so that it really prevents the development of the cancer in the first place, right? And so a lot of these antigen presenting and receptors on the different cells are available and known. Many of these treatments are what are targeting a lot of these. And so what is involved in this study is where they were trying to identify and look at efficacy between two different molecules, 3H3 and SA41BBL. And this project generated single cell plus bulk plus deep immunophenotyping flow cytometry. And so a lot of single cell deep phenotyping and cell annotation was required because nothing really existed. And so what this was is a very complex experimental design where you have these three treatments. There are two injections that are given 14 days apart. There are two time points and samples collected at 3 and 7 days for every injection. And then there are two sites where you have lymph node and the skin where, at each and every place, you have two tissues that are collected. So there's a very complex design for this method. And so what was definitely involved and required is that, yes, we start with our basic pipelines that are analytics, go up to that level. But given the number of comparisons that we were dealing with every time, 48, 50, 56 comparisons that we needed to look at simultaneously, we had to be a little bit more creative and come up with our own barcode and other strategies. I will not go into specific details to this because in your future INBRE webinars, you will have a chance to listen about this work in more detail from one of our data science personnel. And so what I want to give you at this point is just a flavor, a gist of what is feasible. So in doing so, we continue to add more applications. And so what we could do is break down into the different pathway categories and really come up from our barcoding strategy to explain a handful of these biomarkers, which are most essential and important, and explain the differences in the efficacy between the 41BBL and the 3H3. You're looking at the different enrichments and then, importances for the different pathways in reactome. But in doing so, we actually also utilize our own IMPRESS method to really be breaking down into the sequence of the pathways in which these different biomarkers were involved in, which really explain the differences. So as you can see here, bottom is SA41BBL, this is 3H3. The connectivity that goes back and forth between these pathways is really what differs between and explains the differences in the efficacy. And you'll see how many of them land up being common versus unique when you're looking at the different pathways that are involved in these two mechanisms. So the bulk part of this is already published, and you'll see every time a yellow box with the citation in it. We also developed this CIR portal where the bulk part is already functioning and the single cell part is being thrown into it right now, but where users, not only our collaborators, but everybody in the world can go to this portal because there are others working on immunoprevention and they can actually upload their data or they can come and look at the data that this project generated and how it might compare or how they can benefit from accessing these results. So a lot of this is interactive on these portals and that is available and so you'll always see URLs and citations on these slides. Another example in this space that I would like to showcase is this neurodevelopmental changes study with oxycodone exposure. Now, opioid drugs are very commonly given to pregnant women. They do slow motility, gut motility, permeability, and affect the microbiome. And what this study was focused on is looking at mice models who are exposed to oxycodone as to what kind of adult neurobehavioral or metabolic changes are seen and how this sort of translates into the clinical setting. So the data here is really microbiome, 16S RNA sequencing data. There's a bunch of tools used, which again, I won't go into deeper dive right now. But what you're seeing is between the female comparisons and the male comparisons against the control, the different taxonomical bacterial OTUs and others that you see are very different, right? They are very different and they are all significant in terms of changes. And so females and males actually respond very differently to even the same oxycodone treatment. What was integrated here is with 16S RNA, a metabolic profile. And so what this allowed us to do is look at the different taxonomic bacteria, but also the metabolites, which are up or down regulated based on blue or red coloring and the size of the circle and which pathways were getting impacted or affected, right? And again, you can see between male and female, there was such a big difference. So we could pinpoint to which taxonomic differences they were looking at, but also go a step further because now we not only had a microbiome and we had metabolome, but we also had the information from a lot of studies which were neurobehavioral studies. And I'm going to zoom into one of that you can actually see Barnes, Maze, Echo MRI, some predictions coming from these microbiome and picrusty or other sort of neurobehavioral studies that our collaborator was doing, which was explaining the phenotype much more clearly. And so what you could do is you're seeing a positive correlation in a red here and a negative correlation in a red that really links these biomarkers from these different data types along with the different phenotypic measurements, right? Which means it clearly tells you if something is going up, how is it going to impact your phenotype? If something is going down, how is it going to impact your phenotype? And this is something that can really be used so that you can come up with the hypothesis or the next steps in the treatment and how some of these things can be utilized or converted into treatment options. So now let's go into the bottom part of our first pillar, which is really showcasing some things that have been developed in the framework space. And then there are a couple of developments over here. So the first one is soy KB. Again, if you're doing a lot of work on soybean, this is the place to go. Its development started back in 2010. It's been a public resource since then. So we have a good 15-year plus history. It's very extensively published, supports all of these omics plus analytics components. And it all started there because even the USDA databases at that time did not have these capabilities. With our experience in soybean, a few years down the line in 2015, we expanded and built this KB Common system, which now supports each and every and all kinds of organisms. So over the period you can see many different organisms added here. But the main really benefit is that in soy KB, we were the controllers, whereas in this, we actually added new functions so that any user can start their own KB as long as they bring the files that are required in the first 6 basic. Generally, if you have a human genome, you have a genome, you have those 6 files available. They can add and update versions of those. They can contribute their multiomics data to the KBs, and they can then go and browse these knowledge base. KBs are knowledge base for short. All of this is seamlessly connected with high performance computing systems. So a lot of the times data analytics is also supported, but our focus is mainly on data integration. It's got this Laravel framework, MySQL, MongoDB, and a lot of JavaScript, PHP, Python, and other things, mainly focused on visualization components, which is what I want to showcase to you guys. Now, this is what our old version, website version of KB Commons looked like. And I just took a screenshot of what all things were available on the menu. And that gives you an idea. We actually have 6 different things that you can search as entities, which are genes, microRNA, SNP, metabolite, plant introduction lines or phenotypes. And then there were a lot of things you could do under browse or tools where you can select your gene list, you can generate your heat maps, your pathways, and whatnot. There was a whole analytics component plugged in for a lot common types of data analytics. And if you were focused more on cancer, it has the entire 24 cancer TCGA, which is the Cancer Genome Atlas dataset, plus the 1046 CCLE, which is the cancer cell line dataset within the KB Commons, Cancer KB framework. So you can directly go play with it because it's all publicly available. You can pull a gene, you can pull a cancer line, you can pull a cancer dataset and go from there. A lot of visualization there supports you, and you can decide if you want to work with all genes or just oncogenes, tumor suppressor genes, any type of cancers, see how close that set of genes is in terms of PCAs for the based on bulk transcriptomics data. If you're more of a clinician and you wanted to study a particular cancer, there are these summary dashboards that allow you to go to your overall distribution of female patients, histology based, tumor classification based, or individual cancer cases and their details, so that you can actually then jump to the genes and their gene expressions and how they may compare with anything in the population. Now, this is something that's a framework that is more goal-oriented from a clinician perspective, because they can look at all of their 100+ patients together, get a summary and a distribution. And this is something we are expanding as part of our Edwards pilot to support lung cancer capability here at Marshall. Now, our KB Commons was newly redesigned last year, and we are working on a manuscript here, which one of my software developers, Sabin here, is working on. And this is really our new website, which makes it a lot more easier and accessible with all the functionalities or new tools that we keep adding. The 2 new tools that I'm going to mention after this are coming to KB Commons, which were not part of the earlier published manuscripts. And so these are new developments that will be incorporated in our new manuscript as it goes out. You can see here again, you can go browse depending on what plants, diseases, microbes you're interested in. We also focus like some of the tools that we develop, which we continuously keep doing. So coming to the 3D omics within the KB Commons framework, this is really focused and replaces our earlier differential expression suite. This is more advanced. You can choose your transcriptome, proteome, metabolome, or other differential comparisons. Once your data was analyzed and added into the KB Commons, it's available here. So you can choose your cutoffs, you can do your own volcano plots, you can do your own overlaps. If you're comparing 3, 4, 6 conditions together, or you can go down and look at the data individually. Now, what it automatically supports is a lot of enrichment for well-studied organisms, which means if the organism has a gene ontology, a KEC pathway, a reactome pathway, you can support and have your data flow directly into a lot of these. And trust me, a lot of these figures are something that can be saved automatically. Or you can actually have them flow into kind of a PPI network, which is supported by StringDB when your organism has a protein-protein interaction information available in that StringDB database. Now, the second tool is CCMT, and the name is a little mouthful. It's cross-species and comparative multiomics translation tool, and that's because it actually does quite some fascinating things in it. There's a cross-species component, there's a comparative component between multiomics, and then there's a regulatory component that you can utilize. So 3 functionalities. First, cross-species allows you to select 2 organisms, whether 2 plants, mouse or human. You can select the comparable experiment or designs that are already in KB over there. It'll tell you how many are homologs, how many are upregulated, downregulated, so forth, the ones that are non-homologs. And then it'll show you a distribution between them in terms of how many are up or down regulated, or you can click on them to do a further deeper dive. Now, you can imagine if you have 2 organisms, they have homologs, but they may not necessarily all behave in the same fashion, right? So you could have homologs which are both upregulated, both down. Some might go in an opposite direction between organisms, which is expected. And you can look and click on any of these to get the list of those genes and look at their expression in scatter plots or their functions or their pathways or functions from PFAM or KEGG. Now, what you could also do is, because your experiments sometimes may not only have transcriptome, but also do proteome, also do epigenome, or maybe do metabolomics at the same time, you're interested in studying and knowing, does your transcriptome or proteome data tell you similar things? Are they doing similar things in that experiment or telling you something different? In this, what you can do is you can select one organism, you can select the experiments that you want to really go look at, and then you can select whether you want to do transcriptome proteome or metabolome or whatever combination that was supported for your experiment. It'll tell you the number of genes and proteins and things in that experiment that you're comparing. And then again, it'll work in a very similar fashion to some of the earlier things. The third piece, which is the regulatory aspect here, allows you to look at transcription factors. So say if you had a transcriptome data and you're interested in looking at what TFs are shown in your data, you could select an experiment that was already analyzed. It will go and help you identify the number of transcription factors, target genes, the differentially expressed one. It'll take you to the target genes of one particular transcription factor and show you and tell you how they are behaving. But you can also go and generate a network where you could see maybe many of them are upregulated, but there could be some that the TF is downregulating. Plus, you could do an overlap, because if you had multiple transcription factors with multiple target genes, then you want to see what are the common or the unique ones in that. You could use an upset plot like this to directly interactively do all of this on the website. Now, all of this is feasible only because from the standpoint that when we flow your analyzed data into the KB Commons, a lot of these things are streamlined to work automatically on that. Sometimes it might need tweaking if the gene names or something are not completely standard, but since these are systems that are developed in-house by us, it is a lot easier for us to just quickly go and expand capabilities or accommodate other data sets. Let me see how I'm doing. I'm probably going to run over time. So this one, which is we are expanding single cell capability in KB Commons. And so I'll just leave it over there in the interest of time. And then we are also building some things which are tied to our DOE project to support this one, which you'll get to hear from, probably Mohit sometime later in our INBRE webinars. Let me move on to the second one, which again, I probably will have to skip a few ones, but let's go from here. These examples are more focused on plant-oriented, so anyone who is working on natural products or working with other plants in the networks, you probably will be able to relate to this even better. We analyzed 1,000+ soybean genome sequences and identified SNPs and indels, and we expanded with some publicly available so that we are looking at about 44 million positions in this. And the goal for a lot of these developmental tools here was to allow farmers and breeders to go and utilize about, we actually over the period landed up building 10 to 11 tools, each focused on using the SNP and indel information, but from identifying new alleles or for identifying which phenotypes that they affect. So there is something called a SNP Viz. If you do a lot of GWAS type of studies, you can actually flow your SNP information, select a region, interactively overlap the gene annotations on it, select or put your GWAS significant results, and then your phenotype data to really go and see which positions might be affected individually, but this is more focused on a small region of your genome. The acute tool, the GWAS only typically brings you to, potentially giving you significant hits, but they very rarely lead to the exact causative mutation in that gene. And so this is a mathematical based work we did with our collaborator, Maria. And the acute tool takes a lot more detailed, dense SNP information from a whole genome resequencing project and then ranks your significant GWAS hits and then scores them so that not only do you hit that region, but you go to the actual causative SNP in this case. The allele catalog tool was again built based on our 47 million SNPs and indels because the goal of this was in soybean, we have many different varieties, land races, elites, and things like that. And then what we wanted to see is what are the different allele combinations, which means SNPs combinations that a lot of these 3,000 lines have. And so you're seeing the number of lines that support that combination per row. And then from there, we wanted to see the effect or the impact of the SNP and how it relates to or explains the phenotypic differences. And so our collaborators who are farmers and breeders actually use this tool practically every day because they are interested in making these phenotypic changes to increase oil, increase protein, increase yield, or whatever SCN resistance and susceptibility related questions. So they actually go and utilize this for identifying new genes and new SNPs that can contribute to those changes. And this tool, which is GeneVarX, again uses the same SNP data, but in a promoter region to see if changes affect the promoter region and also the changes that affect the copy number variation. So if you're familiar with gains and losses of the two copies of the genes and how it might impact a phenotype, this tool allows you to do analysis and prediction using that because the starting point for this is, again, the whole genome sequence data. So that is what essentially the GeneVarX tends to support. Now, we did add a third one because when our collaborators are interested, they are not only interested in working with one phenotype, right, like yield, but they also want to work with a second or a third phenotype, like same time drought resistance or maybe increased protein, something like that. So what the phenotype distribution allows is they can combine the three phenotypes together, then use a ranking of the SNPs so that it will actually land up telling you what are the regions that you have that you can target? And these are all sorted because you will have multiple genes that would contribute to a phenotype, right? But then how do you maximize and pick the best likely candidate? And so that is what this is using significant p-value calculations and so forth. And then when they identify such a thing, they want to go and use the CRISPR technology to go and knock out or make a mutation at a certain place, right? And when you want to do that, you want to make sure you're not targeting protein regions that are conserved, through homology in multiple organisms. So in order to do that, they really there was a need for building this protein sequence logo so that they can now identify and only target the regions which are not going to be detrimental or really lead to a lot of changes if that's a super conserved position through all of these different species. So that is the reason why over the period we continued adding. And the last one that we added was the MADIS, which was multi-allele discovery, because as you would see from the allele catalog tool is there are multiple alleles that can contribute. But at the same time, if you have 4 or 5 alleles contributing to the same phenotype, how can you tweak the effect of every allele combination and what percentage of phenotype? We are looking at pod color from brown to gray or black over here, and how they may be affected and which are the mutations and the SNPs that would be affecting those sort of colors. And so see what all is possible, right? Simply by starting with a whole genome sequence, resequencing project. And once you have these data ready, there are so many different questions that can be answered simply by churning that data in different ways, as long as you are going to be always application oriented. So the only thing I'd like to mention here is that though we started with soybean, because initially our funding was in soybean by United Soybean Board, when we built these systems, we made them expandable to multiple different crops. And we were able to flow in public data from multiple crops, or we in fact continue to continuously increase and add more organisms, with sorghum being our latest one, which is where one of the paper is currently under review. So with that, I think I want to move to my last pillar. As I mentioned, this is more focused on computational method development. And so some of the tools that I mentioned here will be of interest if you have already generated some data or if you're thinking about some projects down the line. One is called Impress, where we land up taking the entire KEGG pathway as a network. And our goal here is to identify the sequential links between the pathways, which are tied to the experimental conditions, changes, differences in your experimental studies. So if you have multiple comparisons you're doing based on transcriptome or proteome or metabolome or any combination of these, we can take the analyzed data from there. We can start as different seeds. So say you have an experiment where the gene is mutated and you're checking against a control. We can take some of the differential genes from there, throw it into this, fill the gaps with some PPI, and what you ultimately get out of this network hairball is a pruned list of pathways, which is what will explain the connectivity between what's happening in your experimental conditions. So when we applied this to a 60-pair tumor and normal sample from Taiwan for non-smoking females who were getting lung cancer, our focus was on why are they getting lung cancer, right? It's very common in smokers, but these are non-smoker females. What is happening at the molecular level that can explain as to what's going on? And that is where you see these bubbles that I have color coded over here. They are they're pointing to these 5 different pathways which had a significant enrichment. And every box over here is actually a gene which is either up or down regulated in red or green, coming from that transcriptome data that is tied with this experiment, which was able to then explain in terms of what is going up and down and how the oncogenes and the tumor pathway genes are really interacting with each other. Now, we have applied this Impress method since then to many, many things, including mice, studies in plants, other human-related studies, because purely it not only tells you the enrichment of the pathways, but it really tells you the connectivity, and we're able to identify key connection spots, which are not really very clear from just enrichment studies. With that, I want to highlight this second tool, which we are very proud about. This is a method called G2PD. It's again publicly available here, published twice already. The first version was only SNP to phenotype prediction. This is what you're seeing from the second version, where you can take any 6 types of these data sets, any combinations of 3 at the same time. You can compare machine learning tools or our CNN-based model. And the goal is really to use these combinations of multiomics data to predict phenotype. And this one actually allows through a web-based interface to allow automated hyperparameter tuning. It then looks at accuracy, but it also at the end pulls out literature inferences or identifies biomarkers and can do both quantitative phenotype prediction or qualitative phenotype prediction. So what can you use it for, right? So think of scenarios where maybe you're trying to predict a phenotype. Maybe you're trying to predict a phenotype in patients, long-term survival or not, maybe healthy or sick mice. The simplest examples I'm giving so that you can relate back to your own scenarios. And then you can take any combinations, like I said, of 1 single type or 2 data types or 3 data types and build those models. Now, G2P Deep supports a whole lot of that. We were the first platform which allowed deep learning through a web-based portal. And so our first paper was in NAR, second in biomolecules. And right now, Mohammed, who is part of the data science INBRE core, is expanding this to single cell and supporting sex-specific predictions. Again, I don't want to go into detail for that. You'll have a chance to hear from Mohammed in a few, webinars down the line. And really the goal for this is to support how the different cell types in a single cell data can contribute to doing these predictions or phenotypic predictions about prognosis or treatment options better, but also how a female and a male perspective might look different and whether those genes are common or are very different in those predictions between the different sexes. So he's working on a manuscript for this, and we are applying this to our lung cancer Edwards pilot. And so that is what we will have coming soon. Another tool that we are applying to our Edwards cancer pilot is something we published previously, which is called IRNet. It's an immunotherapy response prediction, deep learn, again, graph neural network method. It takes bulk transcriptomic data and was originally built on melanoma or gastric or bladder cancer when the comparisons were made with other existing methods at that time. And what it will take is, it will take your bulk RNA-seq data and tell based on that whether the patient is likely to respond to immunotherapy or not. Again, immunotherapy response is based on, the PDL, the PDL1, PD1, and then the CTLA4 status for that patient, which is something that is regularly checked. And you can see that the pathway connectivities for PD1, PDL1, CTLA4 actually look different, and how their status in all of those three would impact whether they would respond to an immunotherapy drug or not. And so we did a great performance in comparison to other methods, and now we are expanding this to lung cancer for our Edwards pilot. So you will see the results come as the applications are done to that. And what you will see is, in terms of the pathways, there are some that are common in all the three approaches, but also a handful that are unique in those, which really guide whether the patient will have immunotherapy, good response or have side effects and things like that. And knowing that ahead of time is actually a lot better. That brings me to two last pieces that I want to mention, and I'll be happy to take questions after that. Cross-MP, which is another method that's developed. You'll see it on the website. It does cross-modality predictions between single-cell RNA-seq and single-cell attack-seq. So if you have one and is interested in predicting other, you can use the method. Usually single-cell RNA is more commonly available than single attack, but this method can go either way around. And so this has been expanded in some animals and some plant species, but if there's a reason you work with some organisms that doesn't exist, it is still possible to expand that very easily. Plus another one that was published just earlier, end of last year, or in fact, Jan 26, it came out, is the SC plant, which is a method that is really focused on a transformer model that is used for predicting single cell types in 4 different plants over here, because cell type annotation is way more advanced in human or animals, but it is not so well, already available in plants. No such libraries exist publicly. And so gaining from our experience from human and plants, we actually build this method so that it works in plants. And right now my student is working on expanding in a cross species. So if you have data from one species, can you predict the other? And the other way around is what. But then this is again already available. So all you have to bring is a single cell data, and it is good to predict. The last one that we are currently working on is a AI-based data curation portal for vaccine development as part of our ARPA-H project. And so what it really does is it takes data from these virus data banks and protein structure and antigen sources, Protabank and antiviral DB. And what we are doing is we are building a curated database for that, which maybe our other Apex performers who are part of the projects can use with their large language models. And so my postdoc, who is again part of the data science portal, the data science personnel, is building an AI-based tool so that the data can also be accessed and directly curated and incorporated in repository by using these LLM type of approaches. But not just that, in doing so, whenever there is a literature or a manuscript, now he has a whole LLM-based data web crawler so that he can get all the different sections of the paper, not just the abstract or the introduction, which is what typically text minors do, but each and every section of this, so that this can be added to the large language models that a lot of our other performers will be using for doing the vaccine design. So this project is a slightly different than what you've seen typically in everything I talked about right now, but how it is able to leverage some of the AI technology to benefit the data acquisition and the data, making the data ready for LLM type of projects. So I just want to come back to this, making sure, I mean, I covered quite a bit of ground. And again, with the time, I would not have the time to go into details for each one of them. But I think the best thing is going to the URLs, going to the manuscripts, and then reaching out if you have any questions or have the need to apply or utilize any of these. And so the best thing is this is now accessible to the entire network. Now, a lot of this work won't be feasible without a great team and a hardworking team. And so we have our KB Commons and so KB developers here. We have our analytics teams. We have our method development teams, which are more on the AI computational side, and cross-MP for Zen. Our biomedical collaborators, I'm just listing some that are currently, ongoing projects here. Our AI and informatics collaborator, our soybean collaborators, and other collaborators on the plant science side here. Again, INBRE, acknowledging our INBRE grant here, because a lot of these methods are something that we'll continue expanding or applying for needs in our network, and also some of our other funded agencies for things that are ongoing. Plus you have our URLs and to go and take a look at it. And before I end, I just wanted to make sure everyone was aware that our new INBRE website is live from April 15th onwards. And so please take a chance, go and visit it. It has now logically organized sections so that you can easily find information. You will always be able to find our webinar recordings under webinars now, and future speakers information also posted under webinar. And if you're not already a part of it, please use this QR code to scan and join the LinkedIn or the Facebook groups, so that you are well aware of any kind of events and other things as we post it over there. All right, so I think I'm gonna stop sharing my screen here and go back to our Zoom to see. Let me see, I think I've got many windows open, so give me a sec to see what questions we may have. And I'm happy to answer those. Okay, so let me see. Let me start by going to the chat. And please feel free to unmute yourself if you wanted to ask a question or if you put it in the chat. So I see one from KB Chin. Does your proteomics tool include crystal structure or NMR solution structure information, as well as PPI interaction? So at this point, if you're thinking more about the structure information from coordinates and others, we haven't really incorporated that in the protein. The proteomics tool is where it really has the protein abundance levels coming from the mass spectrometry. And so we really are not saving even the mass spectrometry peaks, but rather the analyzed abundance levels that comes in there. And it definitely can expand to PPI. I mean, it can link to protein-protein interaction networks, because we are able to tap into some publicly available information and connect from there. I think you have to follow up with drug target interaction data and simulation model. And that is not something we have incorporated yet, purely because I think none of our collaborations were in that space. I think it is something that could be a potential expansion. Because like I mentioned in the CCLE, we do have some drug information in the cancer cell line encyclopedia. So there is capability in the framework to incorporate that sort of a data, but we just haven't spun out a lot of tools which are focused on that purely because I think that has not been coming through any of our collaborations as yet, if that answers. If you have any follow-up questions, do let me know, and I'm happy to take that. Okay, great. So I don't see anything. Okay, maybe some new coming. Yes. Possible to do multiomics analysis if you have data from different sources. So bulk and metabolomics from mouse models and proteomics from a cell model. Let me give it a thought. Bulk and metabolomics from a mouse model, yes, you can definitely get that. Proteomics from a cell model, I think it may be feasible because I think from the cell types, we are ultimately going to go down to the genes, right? So even if you have a particular cell model, you are going to go down to measurements based on every gene that is expressed over there. And in fact, that would be a fantastic case for, if you think about the potential CCMT expansion where you can now compare a bulk versus a cell type, right? We have done some of that in our single cell work, and when we project that onto bulk transcriptomics, we have done that with some of the tools already. So I think it is something that can be feasible. Definitely can take a look at the data. All right, I see something else. If you have only the 16S sequence and miRNA sequence, can we still use the G2PD website to predict? Yes, I think you absolutely can. At this point, microRNA is supported by G2PD. The 16S microbiome is not a type of a data right now in the 6 data types, but I think it's possible to go and expand. The one thing I wanted to mention about G2P Deep is because it's a deep learning method, generally the way the deep learning methods work is that you want to have a good chunk of data which is used for building the model. And so that is possible to do using your own model or where you have public data and you build the model based on that and then do a prediction using your own data that is generated. So I'll give you an example as to how we built it for the cancer, right? We use the TCGA cancer data. So if we were predicting this for, say, colorectal cancer, we had 1,000+ cases. We use that to build the model. And now every time you have a new patient, colorectal cancer data coming, we can actually use that model to predict. If you have single cell data, even one single cell experiment is really great to build a model because you have 5 and 10,000 cells coming from that one experiment, right? So you don't need, in a similar way, thousands or hundreds of bulk RNA-seq measurements or replicates, right? One single cell experiment generally has way more information and enough information that you need for deep learning models. So it's absolutely possible to do that. Yeah. Okay. Any other questions? Don't be shy. Feel free to unmute. You don't have to put it in the chat. If you want to ask anything, just unmute. No, we are a few minutes over. I just want to make sure we have had time to answer anybody's, last minute questions. Okay, I don't see anything else popping. So what I wanted to say is thank you very much for joining this webinar today. We really are kicking it off, the series. Our next one will be coming on 3rd Monday of May. We will have information circulated at least a week before that with details about the speaker. Once we are done with today, we will be posting, and hopefully our recording is still running perfect, onto the INBRE website. And so stay tuned if you have missed anything or if you want to go back to any of the pieces. And I know there's a bunch of tool names there. Feel free to go and visit the recordings anytime, but otherwise feel free to, shoot me an email, reach out to us on any of the platforms. And then we'll be happy to help answer any questions or any needs as you're planning your next projects, right? Next steps for incorporating any omics, multi-omics data sets, or whether any of these tools are suitable for the data that you may have already generated. And these are also fantastic places to basically tap into the voucher programs and everything that are part of the INBRE, because there could be some things that you can directly apply or be able to, work with the data science core to apply or enable if you've already generated some data. All right, so I don't see anything, and so we'll end the recording here.